Good morning, everybody. My name is John Maslowski, and it's a pleasure to be here. I wanted to thank DebRA for allowing us to come to the conference today and introduce our company. Our company's name is FibroCell Science, and talk about a new product candidate that we're currently developing for a gene therapy to treat RDEB. And what I wanted to give you today was introduction to our company and then an update on our progress and how we're approaching this particular therapy candidate, and what our plans are working through the FDA and working with the advocacy groups, and sort of how this all will come together to a product. So moving into it, we have a forward-looking statement. But I want to introduce you. We're new to the DEB community and wanted to talk a little bit about what our company does and what we plan to do moving forward into a new pipeline. Our company focuses on what's called an autologous fibroblast as a product. So you may have heard a lot during the conference about upcoming cell therapies and gene therapies that can be applied to specific rare diseases, and we're in that space. And what we currently have is a product that takes the patient's own fibroblast cells, which I'll discuss in detail on some slides coming up, and then grows them up, and we use them for an actual drug treatment. And we have some advantages, we think, from taking this approach. A lot of our drug delivery is localized. It's not a systemic treatment like you might have with methotrexate or some of the more systemic approaches for disease control. The autologous cells have a reduced chance of rejection because they're from the patient self. We're not dealing with something that's an allogeneic product or comes from another source. And we have a lot of expertise in this. We've been growing these cells since the late 90s, and we've gone through FDA approvals and done a lot of these cell cultures and have a lot of experience with growing them up and using them for therapeutic uses. So we think we have a very nice entry into spaces to work on more complex and rare diseases. So we're going to use our technology that we have on hand, we own, and we have approved to expand on into more gene therapy applications. And I'd like to talk about some of the things that we use our cells for and then how we plan to expand them to treat RDEB patients. So right now, our focus is on using the cell itself for treating other conditions, such as restrictive burn scars and vocal cord scarrings. We have phase 2 protocols in for both now, and these for patients with the scars that prevent movement of joints after a traumatic fire or electrical burn, or around shoulders and elbows and fingers, and they have problems with pain and itching and mobility. And we're using our autologous fibroblasts to treat those scars now. Another one is in vocal cord scarring. Over years, you have use of voice, or if you have a traumatic accident or even radiation therapy or surgery, you grow these sort of large scars on the edges of your vocal folds, and then they don't vibrate correctly, so you essentially have a loss of voice. And we're working on a condition using these same cells to treat those scars. And then, as I mentioned, into genetic disorders of the skin. We think the cell is well positioned for that because, obviously there's a lot of fibroblasts on the dermal layer of the skin, and I'll get into sort of the applications of that soon. So we do have a product that's approved by the FDA, so we have a lot of experience working with the government and working through an approval process. We're located next in PA, which is right outside of Philadelphia. We have about 50 employees, and we're a public company. Okay? So a little bit about our position, but let's talk a little bit more about the cell itself. So I've mentioned fibroblast cells a few times. If you're not familiar with them, they're the most common cell in connective tissue, which includes skin. So that means it's a good target to use for treating diseases of the skin, including scarring and also the more rare genetic diseases. So they're responsible for producing the extracellular matrix, and that's the lattice that forms in the dermis that gives it its structure, it gives it its tone, and it's produced mainly of collagens, and then there's some other proteins such as elastins that these cells produce to keep your skin structure together. They produce other growth factors and cytokines because, as you probably know, they're very active in the wound healing cycle in addition with keratinocytes. So we find them to be good targets for not only scar repair, but also active wounds as well. We find them to be readily cultured in the lab. As I mentioned before, we've grown fibroblasts for quite some time now. And you can see on the picture of the right, here are a field of fibroblasts we've grown in our facility. They grow, they attach to a bottom of a culture dish, and then they expand out, and you can see them there. And there's a lot of scientific precedence for using them in gene transfer. So a lot of work has been done for years and years about using fibroblasts as test cells for inserting genes and doing other types of work. And so we think this is a good mix for using the fibroblast as a candidate for this product as well. So where is it in the skin? You know, if this is the skin's cross-section, the top part is the epidermis. That's where you would find keratinocyte cells. They lie below the epidermis, you know, right there in the dermal layer, and they're ubiquitous through that entire layer. So they're an ample source to collect just from a simple skin biopsy. We can use that biopsy to grow up into hundreds of millions of cells for therapeutic use. And as you can see in the cartoon shown below, they exist in that extracellular matrix lattice. So they're very important for treating any sort of skin condition and supporting skin. So how do we approach our pipeline? We take these cells, are basically the center of our pipeline and really where we concentrate our entire efforts on. And they go into two separate buckets. One is using the cell itself as a therapeutic agent, and that's where we're using it for burn scarring and vocal cord scarring. And then we're working with a company, very interesting they're a synthetic DNA company called Intrexon. They have offices in Maryland and out in California. And what they center on is creating DNA gene constructs for use in creating therapeutics, not only for health, but they also do it for environmental and food. And you know, they hit a lot of different industries. And we find they're an excellent partner to work with our cell type to create a gene therapy product. So on that front, the RDEB is our lead candidate and is really driving the driving force of our company right now for developing our therapeutics. And we're also looking at some other rare diseases of the skin specifically, and of the fascia and other tissues in that modified space. So how do we go about creating this gene therapy? And I know there's been a lot of talks over the week about different avenues of taking a gene therapy, but really the center of it is taking advantage of this basic biological concept that DNA makes RNA and in turn makes protein, and protein is really the target here that we're after when treating specific diseases, genetic diseases. So what we do is we can take that autologous fibroblast that we work with and use a process called transfection in order to take a gene, a little piece of DNA, put it in the cell, and have that cell now express it, even though it may have had a dysfunctional form of that gene previously. So transfection is a process that we do that. We deliver it using a vector or some sort of vehicle that artificially take this gene and put it inside the cell. And if you hear the term transduction, that's just really a way of doing this using what's called a viral vector. And there's a lot of ways you can take these genes and put them in the cell. I just mentioned viral vector. Viral transduction, as you can see in the picture here, is the act of using a safe form of a virus that's been modified in order to be safe, and using that to actually take its material, place it inside the cell, and then that material will integrate inside the cell, and then it will then in turn produce that protein. So where that's advantages is where the cell might not have the ability to produce a protein properly, this little payload that's been introduced to the cell now can. And there's other methods, microinjection, electroporation, liposuction. We've looked at all of these. But the goal in the end is to insert this target gene into the fibroblast cell so they now produce a functional protein for a target. And in this case, we're, in RDEB, there's an obvious target for us to go after. So how this works from a manufacturing standpoint, if you can see the figure on the left, we basically take a skin biopsy from from the patient, very small, about the size of an eraser. We take 3 of those, usually from behind the ear, but in a lot of cases we take it wherever there's available skin, especially in the rare disease patients. We'll grow those up in culture, and then once we grow them up in the hundreds of million cells, we can actually cryopreserve those cells and save them for future treatments as well. So it's not a one and done deal. And then what we'll do is we'll take them out of cryopreservation, they'll be thawed, and then they'll be brought up into a vehicle media, and then they're shipped overnight to the patient for administration. Along this circle is a large battery of safety testing and also efficacy testing to make sure the product works properly, but most importantly is safe. But you can see that's the product on the left that I've talked about for our scarring indications, but on the right, we've added this, what's called the protein expression engine. This is where we're adding the genetic manipulation in order to create these cells into producing a protein of a desired protein of target. So what we do is we prepare a vector. This example shows what is essentially a viral particle, has a little target piece of DNA in there in the picture. We pack it in using the techniques we learned from our partners at Intrexon, and then it's integrated into the cell. And then in our case, we want to see that material actually integrate to the cell's DNA itself, so when they divide, they keep carrying along this newly formed gene with it. And then it moves over into the culture phase and then administration. So really they sort of interact with one another, being that we've added this genetic manipulation step. So it all sort of comes into the same treatment regime. So specifically with RDEB, I think the biology of the issue is specific to a target protein collagen 7. So we know there's a clear problem case here, that there's either a non-functional or improperly formed collagen 7 molecules that are taken from the cells. It's caused by generally a mutation somewhere in that collagen 7A1 gene, and obviously the symptoms, widespread blistering and lesions all over the body. The solution we'd like to find is to genetically modify a cell to produce collagen 7 that can reestablish the functional anchoring fibrils. So you can see in the picture on the left, those anchoring fibrils are missing, and where the fibroblasts would exist, you see the keratinocyte layer on the top. Underneath the anchoring fibril area would be the dermis. The fibroblasts also play a role in the production of collagen 7, and we think would make a good candidate because of its proximity to the zone between the two layers of skin. So this is the problem solution we would like to use for this application. So what are we developing? So in the end, we're developing a genetically modified fibroblast taken from the patient. So it'll be their own cells. It's a personalized, custom-made therapy for the patient. And then they'll be genetically modified to upregulate collagen 7. So now produce what we, and I'll talk about this in a moment, functional collagen 7 that has the ability to properly anchor the two layers of skin based on the morphology of the protein itself. We know that it has anchoring abilities. So what we would do is we would take the patient's cells, we would expand them up to large populations, they'd be transfected and then saved, and then what we can do is then take those out, ship those overnight to the location of the whatever, if it was a surgery, if it's an outpatient, and then they're actually injected intradermally, so we're not doing this IV. This will be localized, so you don't have to worry about conditions of systemic issues, having cells or protein in the IV of the bloodstream. This will be directly targeted to lesion areas, and eventually, to, hopefully we can do it more in a widespread area, area technique. In our initial trials, we'll be targeting specific blisters and working around the periphery of them. Okay? So what are some of the advantages we think we have with this therapy? Well, we've found with working with the FDA and a lot of our safety data that the autologous approach really is believed to reduce the concerns of rejection of the cells. And we hope, and we'll learn more of this when we go through our preclinical and clinical studies, but the longevity of the cell is really the goal here, so they continue to exist in vivo, that they divide, and that we have longer term expression of the protein. So not just a short bolus and then we're back again injecting in a short period of time. We think that having the cellular engine there modulates some of the way the protein is laid out. So, cells signal one another, they produce this protein. They also help modulate its positioning and how it manifests itself in the body. So we think that there's an advantage of actually having the cells present there, and of course having your own cells present to sort of oversee and act as the foreman of all the work of laying back down these anchoring fibrils. And in our case, the vector we're using is actually integrating the gene into the genome of the cell. We think that's important as well because, as I mentioned before, that translates to longer expression. Hopefully, as we learn more from our preclinical models, the cells continue to divide for at least some time and allows longer expression of collagen 7, so we're not repeating dosing at a short interval. So what progress have we made to date? So we have been working very specifically on this indication with our partners. And in the last year, I've been very happy with the amount of progress we have made, considering we're putting this together for an FDA approval for a license. So these are some of the steps that one would typically take when developing a gene therapy. And in the last year, we've moved through a good amount of them. This one's specific for developing the product itself, and obviously, the basic research is out there, but we have to produce it in a way that we can actually get it through the FDA. So that's how we've designed a lot of our experiments. And you can see here, obviously selecting the gene target was clear. We've created the gene construct. We actually have to synthesize these little pieces of DNA that we can place into the cell. And we've done that and shown some early development work that we are doing that properly. We're constructing this entire, it's a very large gene actually. We've selected our delivery system. That's our viral vector that we're planning to use. And we've done actual runs transfecting genes in the cell. So we have data that indicates in what's called a Western blot that we've not only created the collagen 7 protein, but we've created what's called the trimeric, or the fully constructed version of it, with its proper binding sites are attached using other types of tests. So that was a win to us, because now we know that you can make a fibroblast cell that can't, that would normally not have been transfected, but now it has the gene in it, will make higher levels of that protein. So we thought that was a nice lab experiment that was completed that shows that our basic concept is sound. Okay. At the same time, we have to optimize the cell culture. So although these are dermal fibroblasts and we have a lot of experience with them, these are dermal fibroblasts from collagen 7 negative cells. So we had to go back and make sure that they can be expanded using the same method. So what I mean by that is we put them inside a culture dish and we put media on them, and then they start growing, and then we want to grow larger and larger populations of them. We just want to make sure that we do that in the most optimized fashion possible. And so we run numbers of experiments, and we've had a lot of wonderful partners at hospitals, and I'll mention them at the end of the presentation, who have been very good about opening up their patient populations and donating tissue for us to help develop this process. And that's been extremely helpful in making sure we had the right methods down to produce this. So once all that's complete, then we move to a contract manufacturing organization. We make this off of our site since it's a different product, and we've done that. And then the last two steps, we really have to finish up making our, when I say develop assays for potency and safety, we are essentially making tests to make sure the product does what it's supposed to do. It produces collagen 7 properly, it's efficacious, and also that it's safe. We also want to make sure it's free of any infecting materials like microorganisms. We look at specific types of microorganisms and endotoxins, and just make sure the product is safe. And this is not only the right thing to do for the patient, but it's also required by the FDA approval. So we develop all those assays, and it will be applied to every single personalized lot of material we would create. And then at the end, we tech transfer everything, and we got to run what are called engineering runs, basically dry runs to make sure we know what we're doing, and then the FDA will take a look at that and say, yeah, it looks like you have the right method here, and you're approved to start using this in human trials. So there's the product side of it, and then there's also the clinical development. So we just recently announced about a month ago in June that we received orphan drug status from the FDA on this product. I think that's fairly obvious that the disease state is orphan, but that's a status the FDA assigns to drugs to give you some additional assistance in the approval process. So more meetings, more direct assistance, and it's a really good way to start a development program with the FDA, and we have that complete. We also schedule what's called a pre-IND meeting. That stands for investigational new drug. So whenever you have a new drug, before you go into a human trial, you have to show enough data to the FDA that it's safe, that you've done enough work in preclinical models that it can go into humans. And it's an extensive amount of work that we're currently doing now. But what we do in this meeting is basically meet with the FDA and come to an agreement of what we would need in order to get into human clinical trials. So we're actually having that meeting within the next couple of weeks. And that's us talking to the FDA early and saying, what do we need to do to get this over the finish line? And make all those agreements so you don't go down the road and then all of a sudden find the FDA is in disagreement, and then you're going back, and now we're waiting longer and longer for this product to come out for patients. So that's a step we take to make sure we have a full agreement with them. So we'll hold the meeting. There's also a group at the NIH called the RAC Committee. It stands for Recombinant DNA Advisory Committee. As of now, they still look at gene therapy trials to make sure, because gene therapy is so new, and there's a lot of genetic considerations to be made whenever you design one, make sure they're completely safe for trials, and that the risks have been mitigated as much as they could. So we'll also meet with the NIH as well and just go through that meeting. That's planned. And then we'll run preclinical studies. You use animal models, or there's also skin equivalent in vitro models that you'll run to say, before you're going to put this into humans, we vet this all the way out through these models to make sure it's completely safe. As much as you can, you mitigate all those risks, and then you put a package together, and then you agree with that with the FDA, and you move that through. So we're designing those studies now and actually going to discuss that in that IND meeting to make sure everything is satisfactory for public safety and also scientifically sound, and then we'll move those through as well. And then the IND filing will enable human studies, and then we'll move right into a phase 1 clinical trial. And we've already have partners that we've already discussed where we're going to run the study, who our investigators will be, and we've actually already pre-designed this study and are looking forward to hitting that milestone. So what do we think the next couple of years will look like? So what we're able to do from the industry side is to move this very quickly through the process. So in 2014, we're looking to complete product development. We want to hold our pre-IND meeting with FDA and then initiate the preclinical studies. So that's what the rest of this year will be mainly about. In 2015, we hope to have the IND filed with FDA and then actually starting clinical studies. So we've been developing this for a little over a year now, so we think we've had a nice rapid pace with this development cycle and hoping to be in humans sometime in 15. So just a quick summary on the product itself. We're obviously have a lot to learn about the product over the next couple of next year or so. The preclinical studies, the questions about how often are we going to dose, how much, what's its effectiveness on blisters of various sizes and lesions of various sizes going to be, how the area of injection treatment, all that we're going to answer with our preclinical studies and dose in different fashions in order to learn as much as we possibly can. We think there's an advantage here for having a biological solution for these genetic disorders, and when you're treating a genetic disorder, it logically seems a gene therapy should be somehow involved. And I think we have a nice solution for that. I think that we hope that a lot of the early data we see is very, very promising from at least the laboratory part of it, and it's time to execute that and making sure that works in an animal model. We think, talking with experts and really just kind of thinking about the skin condition itself, a localized approach we think is advantageous, over a systemic approach just on the health and safety of the entire part of the body, including the organs and other areas. Fibroblast cells, big proteins in the bloodstream, that could be problematic. So we avoid that by treating directly to the area of concern. Autologous therapy, we've learned through our long history of developing this and putting it through the regulatory cycles and actually administering to patients and having safety data, that there's advantages over that because of rejection rates. We think the cellular engine is advantageous for actually sustaining deliverable proteins. We think integration in the cell genome, there's a lot of ways to do this, but this one made sense just for the longevity of protein expression. So we have that here, and we'll obviously have a battery of safety testing around that done to make sure that there's no issues with the way we deliver. And of course, extensive testing profile to ensure safety and efficacy. We have batteries of tests that are done in every lot, in every culture that we recreate to ensure that it meets all the standards and for the patient and for the regulators. And if you look in the news, and I'm sure you have and you heard on this conference alone, gene therapies and these alternate biological therapies are just really becoming more and more accepted. You can look at rates of clinical trials for gene therapies are going up. There are a lot of companies working out there working on very rare diseases like Bluebird Bio who are using gene therapy and have had nice progress in their chains and are really where the innovation of sort of medicine is now apart from large pharma. So our commitment to this project, we're a highly focused team. FiberCell is a very small company, but that allows us to focus very specifically on our goals. So we're very highly focused on this, and we're very motivated to help the patients. That's really the number one reason we want to do this. Is rare diseases for us is a very, very interesting place to operate because we think that's where the most patient impact is, and we're really excited to really put a lot of our efforts towards that as well. We're moving into a more medical part of our company, and we're very excited to be able to do that. We have a veteran management team, a lot of people with experience with moving products through, so the goal here is to move this quickly through the approval process. A lot of years have been done in research, but we think we have the ability to move this instead of waiting 10 years, 2 or 3 years, and is more the goal. If we can do this in under 5 years, I think that's a lot better than the long-term solutions. We have access to significant scientific expertise and resources. Our partnership with Intrexon, they are some of the top, molecular cell and biologists and geneticists that we've worked with, and they brought a lot of wealth of knowledge to our company to create very complex products like this. So that has given us a lot of access to technology and to move this forward. We're working with the top key opinion leaders in the field. I'm sure most of them are here. And they've been extremely helpful in guiding us properly, not only through the patient's needs, but also through the science and through the regulatory part of the experience. We've been in communications with DebRA. We think that it's very important that patient advocacy is part of our drive, and that you tell us what we need, to focus on when we develop these products, and how to reach out properly and help with the government as well. And we went to the FDA very early on this. I think a lot of companies where they wait to the last moment to speak to the FDA, and then all of a sudden they learn all these aspects of their program that they need to go back and repeat. And so we felt that, knowing the experience we've had with our previous cell product, that it's very important to start early with the FDA and have those agreements and meeting up front. And I think they actually appreciate it as much as we do, to get the feedback from them. And we're well funded, so this program is paid for through its development, all the way through the phase 1 program, and we think that'll help keep the speed on it, to keep the development moving. So we think that combination of these commitment items, we hope to bring this to the market, a lot more rapidly than some folks have been able to in the past. Okay, so I just wanted to thank a few groups here. Obviously, DebRA, Brett Copeland has been very helpful and has guided us very, very specifically to what we need to do and what's important in the disease area, and has pointed us out to a lot of key opinion leaders that have been helping us. Specifically thanking Stanford University, Dr. Al Lane and Peter Marankovich have been giving us very excellent advice and guidance and how to help navigate through this field, and has been very valuable for our programs, our rapid progress. Cincinnati Children's, Dr. Ann Lucky is one of the first people I talked to, and it was wonderful in really explaining the patient's needs and telling us, how to help us really understand the area. And Dr. Richard Aziz Khan has been helping us actually collect tissue for our development work from RDEB patients, and have been invaluable as well. And of course, our partners at Intrexon, who are scientific leaders in the area and have been very helpful with us moving the program forward. So if you want to learn a little more, our website is fibrocellscience.com. You can learn a little more on the company, and we'll obviously post our progress and notes on how we're doing. I hope next time we speak, I have a lot more. I have actual data I can start showing you, and actually in preclinical, and hopefully maybe even some human data by then. Hopefully we've progressed enough. And again, I want to thank DebRA and everybody for listening. Thank you very much.